Okay, today's lecture is about transposons. They are some people make careers out of studying transposons, and then you will see why they are so important. You could think, oh, maybe they make up genes that are essential to our existence. Maybe. So we'll see how they were discovered, which might actually be my favorite part about transposons. And then you're going to see that there is a big variety of them, and it's, for me, the biggest part of like that big variety of them is to know that there are different types, but the two types I want you to remember are retrotransposons and DNA transposons. So if you don't remember nuances of each of them, what I want you to remember is that there are these two, and then you will see what's the difference between the two of them. So, and there are, as I said, different types of transposons, but first slide, as always, is what we wanted to learn from this, and then we're going to jump into the history. So I want you to know, what does that mean? And in the very last slide of this lecture, I tell you about the C-value paradox. So you will learn of what a C-value is. You will think, what's our expectation about the C-value? But then you will see that our expectation is not met, which is where the paradox comes in. There are different ways that transposons move around, and then when they do move around our genome, what happens to the phenotype? So why are transposons important to medicine? And then have an idea how prominent transposons are in different organisms. Mostly I wanted to be able to interpret, if I give you a pie chart, then what fraction of that pie chart are transposons. So I wanted to look at a pie chart and say, oh, these names here, they're types of transposons. And then this part of what's the impact of a transposon inserting itself into a gene, that is related to that. So the reason why I bring this up earlier is because of the history of transposons. And sometimes they can be beneficial. People are still studying if they were maleficial, if they were detrimental to us. Why does our genome still carry so much of it? All right, so here's some background information so that you can understand how transposons were discovered. So over here on this picture on the right, there are two different corn kernels. So just imagine, Thanksgiving, when you have those colorful corns that you can buy, and mostly for decoration. Each kernel came from the fertilization of one egg. So those hairs from the corn? That is the style of the flower. That's like the female part. That's kind of that's the tube through which the pollen travels and then fertilizes the ovule, the egg in that female flower. And then once that fertilization happens, then it fills in as a kernel. So let's think about this. I have this kernel here that's white, this other kernel here, they call it purple. And let's think about their genetics. So this could have an egg that has genetic composition different from the egg of this other kernel. There were two different ovules. So two different female cells that were fertilized by gametes. And then one gamete fertilized this one egg, the other gamete fertilized this other egg. All right, that's the background on how you get the kernels. And now the color, let's say that it's conditioned by this white allele. So if you don't remember what alleles are, I'll explain. You can raise your hand. But it's a variant of a gene. So the gene here is kernel color. And we're going to say that the allele that conditions white kernels is the big W, and it's dominant because it prevents the formation of the purple color. Okay, well, that's not why it is, but this is how it does it. It's dominant because if you have the heterozygote, which would be big W, little w plus, then that heterozygote will still be white. That's how you know that the big W is the dominant allele. All right, so big W conditions white, little w plus conditions purple. But if it's recessive, it has to be in double dose. So corn is diploid. So if all of the alleles for the locus, in this case you only have 2 alleles because diploid. If it's this, then you're going to have purple kernel. So if you're paying attention, now you actually already even know the answers to these. I'm going to do to write a poll for the top one, and then maybe I'll do another one for the bottom one.